Welcome to the third video on course on cell cycle. Here we will learn about MPF, its structure and how was it identified. So in eukaryotic cells, progression through cell cycle is controlled by a series of protein kinases, and that has been conserved from yeast to mammals. But how was this key factor identified in cell regulation? Now, initially, distant experimental approaches contributed to the identification of the key molecules responsible for cell cycle regulation, and these were three independent experimental approaches. So, in the first approach, it was seen that in a normal hormone treated frog oocyte cell, when hormone treatment is given, the cell progresses from G2 to M. When the cytoplasmic factor from such hormone treated cells, they were injected into the cells that has not been exposed to hormone, they also triggers the transition from G2 to M phase. So the entry of oocyte into meiosis is frequently referred to as oocyte maturation, and this cytoplasmic factor, this was hence called maturation promoting factor or MPF. MPF was later seen to be also present in somatic cells, and thus MPF was appeared to act as a general regulator for transition from G2 to M phase. Now, the second approach was identification of protein kinases and cell cycle regulation. It was with the help of genetic analysis in yeast, temperature sensitive mutants were identified which were defective in cell cycle progression. So the CDC, which stands for cell division cycle mutants, they underwent growth arrest at specific points in the cell cycle. For example, mutation at the gene CDC28 caused the cell cycle to arrest at start. Hence, the protein encoded by this CDC28 gene, the CDC28 protein, is required for the passage through this critical regulatory point at G1. Similarly, CDC2 arrest Saccharomyces pomni cells both in G1 as well as in G2 to M phase. Later on, it was identified that both the CDC2 and CDC28, they encoded a protein kinase known as CDK1. In the third approach, it was seen that two proteins were identified which displayed a periodic accumulation and degradation. It accumulated throughout the interface and it started degrading after the entry into the mitosis. Hunt in 1983 called these proteins cyclines because of the cyclic appearance and disappearance, cyclin A and cyclin B, and suggested that they might function to induce mitosis. Microinjection of cyclin A into the frog oocyte is sufficient to trigger the G2 to M phase transition, which was shown in later days. So, when these three experimental approaches converged in 1988, that led to the discovery of the MPF structure. So, MPFs were purified and shown to be composed of CDK1 and cyclin B. So, CDK1 has the catalytic activity and cyclin B has got the regulatory activity. So, cyclin B is the regulatory subunit required for the catalytic activity of the CDK1 protein, and together they are known as the MPF. So, in this video, we have learned about what is MPF, how was it identified, what is the nature and structure of MPF. So, in the next video, we will learn about mechanisms of MPF regulation. Thank you.